So welcome everybody to today's webinar on exploring human disease and protein variant data in UniProt and contextualizing variation with ProtVar. I'm Yvonne Lucy, I'm a senior database curator at UniProt EBI, and I'm here with my colleague James Stevenson, lead data scientist at UniProt EBI, who will present the second part on the ProtVar tool. In this webinar, you will learn how to navigate UniProt KnowledgeBase to find and discover information on proteins involved in human disease and disease-associated protein variants. I will show you how to search for specific diseases or a protein variant in UniProt, how to visualize a disease variant, how it may affect protein structure and/or function. I will show you external disease-related resources, the links that we provide in UniProt, and how to access and download natural variant information and data sets. And in the second part, my colleague James will talk about ProtVar, a tool to contextualize protein variation with protein structure and function information. So what is UniProt? UniProt aims to provide a comprehensive, high-quality, and freely accessible resource for protein sequence and functional information for a large variety of species. We are providing primary sequences for proteins, including isoforms, variants, and post-translational modifications, physiological protein functional information, structural information including topology and access to 3D structures, both experimental and predicted structures, as well as data analysis tools such as BLAST and multiple sequence alignment tools. And importantly, we provide stable identifiers, UniProt accessions, to help identify specific protein sequences. So what kind of data can you find in UniProt? So we provide the protein accessions and sequences, and for every protein entry, or for the protein entries, we provide protein functional information, including enzymatic reactions, as well as subcellular location information, protein-protein interactions, 3D structures, protein variants, and for human proteins, we also provide involvement in disease descriptions. This information is coming from the scientific literature and is annotated by our curators at UniProt in a manual process. But for protein variants, we also import data from other resources, such as ClinVar and large-scale data sets from Ensemble, Cosmic, and GNOMAD. So when you come to the entry page of UniProt, you will find following information. First, you find the free text search bar, where you can find your protein of interest. The default search is set to UniProt KnowledgeBase, which is one of our data sets, and this is made up of the withProt section, which contains all the reviewed protein entries. That means they are manually curated by our expert curators. There's also an Tremble section, which contains the unreviewed protein entries that have not been touched by our curators, but they contain annotations based on automatic annotation tools that are based on the manually annotated information in withProt. Then we also have other data sets, such as the proteomes, the protein clusters in UniRef, and UniPark, the sequence archive. But we will look at the UniProt KnowledgeBase today for the human protein variation data. Then at the bottom of the UniProt entry page, you will also find the analysis tools such as BLAST and Align. I will not go into detail on those today, but you will find a recorded webinar on our training page, as shown here on the bottom left, where you can learn more about using BLAST and Align. We also have a ID mapping tool and a peptide search tool. Then, importantly, we also offer an advanced search. And in the advanced search, you can specify what you are looking for, either by choosing in the drop-down menu the protein name, that you're looking for, or gene name. You can look for a disease, and you can look for a specific variant that you're interested in by using its dbSNP identifier. You can combine these searches in the advanced search by using AND or you can exclude information by using the NOT qualifier. Importantly, at the bottom of the entry page, you will find supporting data, and there we have a link to the human diseases that are annotated in UniProt. And when you access this tab, you will get to the human disease list that are described in UniProt. Currently, we have 6,740 human diseases described. And to look up a disease that you're interested in, for example, Alzheimer's disease 3, you can click on the disease identifier. This will bring you to the disease page. Here you will find more information about this particular disease, for example, the definition, which is based on the definition by the Mendelian Inheritance of Man database that is establishing the gene phenotype relationships. You will find the acronym for the disease, synonyms, keywords, as well as cross-references for other clinically relevant databases, such as the mentioned Mendelian Inheritance of Man database, MedGen, and MESH. And importantly, below the disease description, you will find related UniProt entries that contain proteins that are associated with this disease. For example, here, the human presenilin 1 that is involved in Alzheimer's disease 3. For this protein, you can see a quick overview on the information that is annotated, such as the variants, isoforms, and other information for this protein entry. Then you can click on the UniProt accession to get to this specific protein entry. So in the start page of human presenilin 1, you will find information such as the molecular function of the protein that has been manually curated from scientific literature. And you will also find the links to the PubMed publications that you can look up for more details about this information. On the left-hand side, you can navigate the navigation tab to other annotation type sections, such as interaction information for this protein, structure information, or family and domains. But we are mainly interested in the disease and variants information for this protein. So in the disease and variant section, you will find all the diseases that the protein is involved in. So we will find that human presenilin 1 is involved in Alzheimer's disease 3, but you will see that there are more diseases listed for this protein entry, such as frontotemporal dementia 1. For every disease, we list the variants that are associated with this disease below the disease description, as shown here in this table. In the disease description above, you will find a note that explains the role of the gene protein in disease pathogenesis. So either causative susceptibility or modifier gene. And for this disease, for this protein, it says the disease is caused by variants affecting the gene represented in this entry. So this gene is a causative gene for Alzheimer's disease 3. Below the disease description, you will find the link to the MIM database. And by clicking on it, it will bring you to the site of Mendelian Inheritance of Man database, where Alzheimer's disease 3 is described in more detail. You will find the phenotype gene relationships and other genes that are involved in the disease, such as ApoE. You will find the description of the clinical phenotypes and other variants listed that have been associated with the disease. So as I mentioned, below the disease description are the natural variants listed that are involved in Alzheimer's disease 3. And here you will find information such as the UniProt variant identifier, the variant position, the amino acid change, and the description for the variant, including the disease association, the variant impact description, if available, including the link to the publication that describes this variant impact, or the variant identification, and we provide also the dbSNP identifiers if available. You can click on the dbSNP identifier link, and that will bring you to the short genetic variations database, where you will find more information about this specific variant, such as clinical significance, as well as frequency in populations. So for human protein variant data in UniProt, we capture mostly human missense variations, and we have currently over 83,000 missense variants annotated in UniProt. And thereof, over 32,000 variants are involved in human disease, meaning that they have been classified as pathogenic. You can access this human missense variation data set in the FTP download, and find the list of missense variants in the HUMSAVAR text file. So in UniProt, when curators annotate a variant for a human protein, we classify the clinical significance of this protein variant by using the guidelines by the American College of Medical Genetics and Genomics and the Association for Molecular Pathology. This categorization is based on findings described in the scientific literature, such as family history, the molecular impact of the variant on protein function, as well as conservation of the residue and prediction tools. And with this guidelines, we then classify the variants into benign or likely benign variants, variants of uncertain significance, and likely pathogenic and pathogenic variants. Just to mention shortly that we have tools to analyze the variant impact on protein structure and function, and I will talk more about this in the next few slides. We provide the feature and variant viewer in UniProt for every protein entry, where you can visualize the overlap of variants with protein features, including structures from PDB and AlphaFold, and then the before mentioned BLAST and align tools to allow you to analyze orthologous proteins and look at the conservation of the residues involved in the variation. And we provide the crosslinks to relevant databases such as OMIM and SNP database and PDB and ClinVar and AlphaFold. So going back to the human presenilin 1 entry, we have been in the main entry page, and with the tabs here at the top, you can navigate to the other sections of this protein entry. So here we are in the variant viewer. In the variant viewer, all the variants associated with or found in this protein are shown in a variation matrix along the protein residues on the x-axis and according to the amino acid changes on the y-axis. In this variation matrix, you can filter the variants for their consequence. So the pathogenic variants are shown in red, the benign variants are shown in light green, and the ones with uncertain significance are shown in dark green. And the blue ones are mostly variants imported from large-scale data sets, and they have you can look at their consequence by prediction tools. You can also filter for the provenance of these variants, either looking only at UniProt reviewed variants that are curated by one of the curators at ClinVar variants, or filter for large-scale study variants. And this data can also be programmatically accessed via the UniProt Proteins API. which you find at the bottom of the UniProt entry page, where you can get the natural variants by UniProt accession, dbSNP identifier, or HTVS expression, or look for variants related to disease, location, source, or consequence. In the variant viewer, below the variation matrix, we also show the variants in a table format. And by clicking on a variant line, you can expand the variant line to see more information on the disease association, the pathogenicity, the source of the variant, and cross-references to other databases that are describing this variant of interest. You will also find the genomic location. And up here, the different variant identifiers from the different databases, the amino acid location, amino acid change, and the characterisation, as we have seen before. And yes, the pathogenicity, including the source with the categorization of pathogenicity. So then we are moving to the feature viewer by using the tabs at the top of the entry. And in the feature viewer, we display all the protein features that have been annotated for this protein. So this includes the topology, domains of interest, sites, post-translational modifications, and other information such as mutagenesis assays. And we include the variants here again, here shown in the variant track, the number of variants, and you can click on it to expand the variant track to the variation matrix, as we have seen in the variant viewer. I just would like to briefly mention that we also show the AlphaFold confidence scores in the feature viewer, as well as the alpha missense scores for the average residue score, as well as when you expand this alpha missense track for the individual amino acid changes as a prediction pathogenicity for each residue change. And again, this data can be accessed programmatically in the Proteins API in the feature section, and you can get the features via UniProt accession or feature type. So here is the feature viewer with the features at the top, and then the variant track expanded to show you the variation matrix. And I have selected a variant of interest, which is the variant at position 435 with an amino acid change from leucine to phenylalanine. And you find the description of this variant here, including cross-references and the publications that are describing this variant. And you will see by selecting the variant, it will bring up a light yellow line that indicates the overlap with the features above of the variation matrix. And you will see that our variant of interest is lying in a motif which is named the PAL motif. And this indicates you that this variant may have an impact on protein function by changing this PAL motif. And you can read up on the more about this motif in the publication mentioned here. In the feature viewer, we also provide the protein structure, and this is below the features and variant table. Here we provide structures from PDB, experimental structures, as well as AlphaFold predicted structures, and you can filter for the source here or then for the method by either looking at NMR structures, EM structures, or predicted structures. And if you again select a variant in the variation matrix above, it will then highlight this residue at the variant position in the structure in yellow, and it allows you to zoom in to see more details about the surrounding structure for this residue of interest. So to end, I would like to mention again the different possibilities to access this data in UniProt. So for the human disease and variant data sets, they are accessible in the FTP download. And for the large-scale variant data sets, they are accessible in the Proteins REST API, including their genomic positions. And with this, I would like to hand over to my colleague, James.
Yvonne ended there on talking about programmatic access. And I know at least one person in the chat has asked about programmatic access. So I'm just going to start with that before I start talking about ProtVar. So if I just show you an example in Colab Notebook, hopefully then you can get an idea of what programmatic access might look like in UniProt. So of course, you can just write this in Python on your own machine. This is a Colab notebook to show you kind of what it looks like. So this is kind of the way you would set up accessing the API via Python. And you can see, and I'm going to share a link to this at the end so everyone can play around with it and change it at the end. So this is briefly how you would set it up. You can see here we're pointing at the Proteins API where all the data that Yvonne was talking about is located. And then once you've hooked up to the API, you can see you can write things just as you would do with your own data in Python, and you can produce tables, but the data is directly from the UniProt Proteins API. So the API itself, if you want to look at it directly rather than interacting with it via Python, you can go to this URL, which will be shared with you. And the one that you probably care about most in here is the variation part. So you can see on the left here, we have variation. And in here, you can type in either certain positions, or you can type in a whole accession, and then you'll be able to get, I'll put an example here. If you type in accession and try it out, then you get all the information that we have on variation in that protein. And of course, this is a JSON object returned, and you can parse that yourself. Okay, so you can ask questions about that at the end if you like. And also, I'll send you a link at the end so you can try the Colab notebook out for yourself. But the thing I mainly wanted to talk about today was a resource called ProtVar, which will help you contextualize human missense variation. So of course, Yvonne talked about a lot of different types of variation, and actually in many different species in UniProt. But I'd like to talk to you specifically about missense variation in humans. So much narrower, of course. There are roughly kind of 5 types of variation: structural, single nucleotide polymorphisms, insertions, deletions, copy numbers, translocations, and inversions. But I'd like to talk to you today about the single nucleotide polymorphisms, and specifically the ones that lead to an amino acid change. They are the most common by far of the variants in humans. This is probably an underestimate. 90%, maybe as many as 99% of the variations in your genome will be SNPs. Of those, that's about 4 million per person, 4 or 5 million. There's a lot of variants. 9,000 or so will be missense variants. That is, 9,000 will be single nucleotide polymorphisms that occur in coding regions in proteins and change the amino acid identity. So what is ProtVar, this thing I've been talking about? It's a variant-centric collection of relevant annotations. So rather than UniProt, which shows you the whole protein, and then you can get down to certain regions or certain positions, ProtVar is entirely on one amino acid position at a time. It maps variants from different formats and versions. It then retrieves and organizes all the data you might need that are relevant to understanding and interpreting your missense variant. As well, it has novel predictions that are just in ProtVar to help you with interpretations. And this is it's been released about 1.5 years now, but it's already well used across the world by a whole range of different people. And if you want to try it, this is the URL, and you can also scan this QR code. Okay, so the first part of ProtVar, the really critical part, is the mapping between different formats. So in order to bring all this information from different places, mapping is entirely critical. So you can see here some examples. So Yvonne talked about the DB SNP ID. So this would be an example of a DB SNP ID. And we map all of the DB SNP IDs to GSH38 genomic coordinates, but also cosmic IDs, ClinVar IDs, also cDNA or protein positions. These are all ways that you can enter ProtVar. So if you have a list of variants, perhaps you've got some cDNA variants, perhaps you've got some protein positions or IDs, you can add them all into ProtVar and it will map them all for you. We also do this GSH37 or 38. You can enter your variants in either 37 or 38 coordinates. If you don't know which reference assembly you have, then ProtVar will detect it for you and do the assembly for you. We also map all the way through to isoforms, PDB structures, and AlphaFold structures. And we have complete mappings for over 93% of human proteins. And because we have pre-mapped all of these things, these are almost instantaneous. So I have got some static slides, but I'd much rather show you what ProtVar actually looks like. So I'm going to be brave and press this. Okay, so this is what the ProtVar entry page looks like. So you can see here, either you can add your variants into this box, and here are some example genomic variants. You can see the formats are quite different, but these are all accepted by ProtVar. Perhaps you have some cDNA, and you would enter it here, or protein, maybe you have an accession and a protein position rather than genomic, and you can enter in that way, or variant IDs. You can see down here, we also can auto detect the build that you have, or you can state which build you have. If you want to search for a whole protein, you can also type your UniProt accession down here, and it will retrieve all the information on that protein for every single possible variant, not just the variants that we've seen before or we have, but for every possible variant for your protein. So just to give you an example, I've put 4 genomic coordinates in here. And then when I submit, you can see this is very quick. As I said before, we've pre-mapped it. So that means if you put 1 million variants in that box, it will load in a similar time, certainly under a second. And you can see here, we've entered these genomic coordinates. It's mapped it to the protein. It's got the codon position and whether it's a positive or reverse strand. We have a prediction here from CAD, and then we've mapped to protein position, protein accession rather, and position, and the amino acid change. So this is kind of the basic mapping of ProtVar. But really where ProtVar, I think, is even more powerful are these 3 buttons at the end, which show you annotation specific to your position. So for example, this last one, we have a histidine to arginine change, position 493 in this accession. And what we see here is that we have information from UniProt, which shows a 3,000-fold reduction in activity here, 15,000-fold reduction in activity there. And if you click there, you can see the papers where that actually comes from. Down here, we can see some predictions from alpha missense, from EVE, ESM1B, and CAD. In this particular state, they're all happen to be deleterious, but of course we can be mixed here. We've got some stability change, which I'll talk about later. And then we have a conservation, which we calculate. And you can see on the right, this is information about the region surrounding the variant. And of course, the variant may interfere with a function in a domain. So we can see in this instance, we have a predicted pocket, which this variant is in, and we have some confidence. I can explain that later. And you can see also another unique thing to ProtVar where we predict protein-protein interfaces. And we can see in this case, this particular protein amino acid is at the interface with this protein. So we can also look here at co-located variants. So when I mean co-located variants here, I don't mean at the same genomic position. In this case, I mean at the same amino acid position. So you can see this is a histidine to arginine change. And as Yvonne showed earlier, these are all the different databases that particular change is found in, and we can of course link through to them. But you can see also there is a histidine to leucine change seen. So this might not be the same genomic position, but it is in the same amino acid position. And this can be particularly useful if we have information, disease information, for example, in a variant at the same amino acid position, but we don't have disease information at the variant that you have submitted. So I'll go back to the presentation, and we can skip over these now because I've shown you live. Okay, so I think probably even more exciting than the annotations I showed you is really thinking about how we categorize missense mechanisms. Like, how is the missense variant altering the protein? And the way I like to see it is to split these mechanisms into 5 broad categories. You can see the first very broad one is altering folding. This could be anything from breaking bonds, steric clashes or voids from changing the size of the side chain, changing the torsional angles by inserting a glycine or proline, typically, or gaining or losing charge or hydrophobicity. So that's affecting the protein structure. But you can also, of course, directly alter function. And this could be something like affecting the active sites. So really messing with the enzymatic chemistry of a protein. Or you could interfere with ligand, small molecule, or nucleic acid binding by varying a position. Protein-protein interfaces can be affected as well by missense variants. And we can also disrupt PTMs. So in ProtVar, we've calculated using FoldX 208 million stability predictions. We've used AlphaFold 2 models in the high confidence regions of AlphaFold 2 models to change every amino acid into all the other 19 amino acids, and then predicted what that would do as far as affecting stability. So you can have all of these 208 million predictions from our FTP site, or of course, if you upload your variants, then you can retrieve the predicted stability change from ProtVar. So secondly, we've predicted 134,000 protein-protein interactions. So these are proteins that have been shown experimentally to bind to each other. But what we have done is to predict where the interface actually is, which amino acids are involved in the interface. And you can see a picture on the right here. This is a picture directly from ProtVar, which you can retrieve yourselves. And you can see here your variant in yellow, and you can see the interface in the first protein, interface the second protein, and you can see that your variant is very close to the interface, and therefore may affect the interface, either overstabilizing or destabilizing this interface. So we have confidence scores for every one of our interface models, so you're able to also filter just the most confident models. And finally, from the predictions, we've predicted protein pockets. So this is areas of proteins we consider likely to be small molecule binding regions. We have 103,000 of these pockets, which we've refined, and these are on AlphaFold 2 models. It's based on AutoSite, which uses physicochemistry and shape to consider the most likely binding site of ligands. And we've created feature points for lead optimization as well. And you can filter these pockets according to confidence. The final thing is we've brought in AlphaFill models. If you're not familiar with AlphaFill, it's not produced by us. The reference is at the bottom here, but AlphaFill aligns experimental structures which have ligands co-crystallized with AlphaFold models. And by doing that, we're able to put the ligands in approximately the correct place on the AlphaFold structures. This is, of course, very useful for validating our pocket predictions. As you can see here, there are 2 ligands in this predicted pocket near to a variant, but also in interpreting variation. See, again, in this case, you can see a variant in a pocket extremely close to a ligand. So you may infer from this that the variant may affect this ligand binding. So you can find all of these things. I'm going to click again and go back to ProtVar. You can find all of these things in the final of the 3 annotation tabs in ProtVar. So again, using the same example, this 493 histidine to arginine, on the right here are all the experimental models, of which there are quite a few, all the experimental models which have this variant position. Not all of the models for this protein, but all of them with this particular position. And if we click here, we can zoom to where the variant is. But perhaps more interestingly and slightly differently to what Yvonne showed in UniProt, we have the AlphaFill models here, and we can also highlight our pockets. So this is the pockets I showed you earlier. You can see the variant in the pockets, and we can zoom in to see that. And the variant is in yellow there in the pockets. And finally, at the interfaces, if we click here, we can see where the variant is. And if we highlight the interface, we can see the interface here and the position of the variant in relation to the interface. You can see in this case, the variant is fairly far from the interface. The reason we show a couple of amino acids back from the direct contacts is that it's been shown previously that the shell behind the interacting shell can often alter the binding properties of the contact residues, which is why we have that. So a variant in this shell behind may well affect the binding interface. Okay, so access, we have a dedicated ProtVar API, which you can use. I'm not going to show you that today, but feel free to go and browse and have a look at that. We have an FTP site, which has all of our predictions in. And if you were to submit thousands, even tens of thousands of variants to our UI, you can also download all of that information. Everything you see, you can download as a TSV, so then you can play with it and filter it later. And if you want to link directly to ProtVar, you can use this kind of format. And using this direct linking, if you were to delete each shell here, it would just shows you more information. This is an arginine change, but if you were to delete that, it would be any of the serine changes. And if you were to delete the position, it would show you all of the variants in this accession. We have sharing features. So if you have a session with your variants from your research and you want to share it with a colleague, you're able to press this and then you're able to share all of the results. If you want to share the downloads, you can also do the same thing in the download section. We have comprehensive help, which is available throughout the UI, and there's also a help section to help you as well. And we're constantly updating ProtVar. So the last update was in February of this year. So really what ProtVar tries to do is to bring together all of the things you can see on screen here. So obviously structure and AlphaFold plays a key role in all our predictions that we have. But thinking about how pathogenicity predictions, human conservation function, mechanisms, and conservation can all help you with understanding disease. So there's an acknowledgement at the end for UniProt as a whole, but just the ProtVar team itself, Prabhat, Tanushree, and Maria in that. And then we also work with our predictions with Zurich and King's College. And our funding, again, is slightly different from UniProt as a whole, including the UK Human Functional Genomics Initiative and Open Targets, where we work with pharmaceutical companies. And again, if you want to check out ProtVar, and I encourage you all to do so, please go ahead. And then if you want to email me directly about ProtVar, then you can also do that. I promise you to give you the link to the Colab notebook, and that is on screen now. So you will get these slides, I'm sure, afterwards. But this is the QR code to go to it, and this is the Colab book for the examples that I showed last time. Yvonne, do you want to take over from here for the UniProt funding?
Yes, thank you, James. So I just would like to mention our funding bodies at the end that make our work possible. And this is the National Institutes of Health, the European Molecular Biology Laboratory, EMBL, Open Targets, and the Swiss State Secretariat for Education, Research and Innovation, SERI. And on the next slide, yes, this is the whole UniProt team, which is divided across 3 institutes, as UniProt is a consortium made of EMBL-EBI, the Protein Information Resource PIR in the States, and the Swiss Institute of Bioinformatics SIB in Switzerland. Yes. And then I would like to mention you find on our webpage information on, for example, the next live webinar, which will be on navigating UniProt, a brief overview and recent updates. I would recommend you to listen to on the 8th of May. Then we have also a lot of tutorials and videos for you to look at on our website to learn more about UniProt and specific topics that you could be interested in. And in the Help Center, you will find documents that explain the different UniProt data sets, how to find things on UniProt. There's some training information and also information on the programmatic access. And please follow us on social media. for training updates and new features. Yes, and as I mentioned, there's the upcoming webinar on navigating UniProt on 8th of May, and there's another webinar planned on April 23rd from Open Targets platform and the genetics of target discovery, which I may listen to as well. And you will find all the recorded webinars from the past, as I mentioned, for example, the webinar on BLAST and the line tools on our training site.
Great. Thank you very much, Yvonne and James, for the webinar today. And thanks everyone for joining in. So there are a few open questions here in the Q&A, so I can read them. I think the first one is for James. So there are two questions within this one. What is the confidence level of predictions shown in ProtVar? And how does ProtVar help in exploring variant co-occurrence or epistatic interactions?
Yeah, thank you for those questions. They're great ones. Yeah, so in the first one, what's the confidence level of predictions shown in ProtVar? So for each of the predictions we have, it shows a different confidence, and we have different confidence metrics for each. So for example, in the protein interactions, there's a certain confidence metric that we use, and we have a filter that you can just get the high confident or low confident ones. You'll be able to read about exactly how we produce that in the paper from that. Also, the stability and the pockets, again, they have their own metrics for confidence levels. So yeah, it depends. As far as the second one, it's really exciting. What your question, and it's something I would love to do. Yeah, co-occurrence is really exciting. Epistatic interactions are fascinating. It's something we certainly plan to do in ProtVar in the future, but we haven't got that yet in the production one.
Great, thank you. The next one is, I think, for Yvonne. How do I link the variant data, for instance, PSEN1 data from UniProt to proteomic and transcriptomic data sets to see their impact on gene expression?
Yes, so in UniProt, we don't capture proteomics and transcriptomics data sets. We don't link those to protein variation. We capture gene expression or impact on protein levels. If they are described in scientific literature, you would find them in the variant description in the manually curated part. But yeah, I'm afraid this information is limited in UniProt.
Okay, thank you. And then another one, I think, is for ProtVar. Can we restrict viewing variants in some domain?
Again, definitely on our list. We can't at the moment. So at the moment you can just search by protein, but we're going to expand that to gene, and it'd be great to do domains. Initially, we'd like to be able to show you all of the information on domains, but eventually what we'd like to do is combine information on domains. So we transpose it from different homologous domains. So that's definitely our direction of travel. But if it's something you're already interested in, please get in contact with us. Perhaps we can work together on that. And if it's important for your work, then we can prioritize that particular piece of work.
Okay, so one question has come in just now. Is it possible to compare protein mutations to ortholog sequences of model organisms?
Yeah. Yvonne, is there anything in
Yes.
you can add to this?
Yes, I didn't include that slide, but I should have, because it's a good question. That so you can identify the orthologous protein using BLAST, and then using the alignment tool, you can visualize the conserved residue in model organisms. So for example, for the human protein, you can visualize it compared to the Drosophila melanogaster protein. That is a model organism to study dementia, and then you can see the conservation of the residue in the model organism on UniProt.
And again, it's another thing that the next thing we want to add in ProtVar in that region is to get the experimental structures from homologous proteins, so you can see your human variant position or equivalent position in model organisms. Because what we find is quite often in a model organism, there's a really good crystal structure in instances where there isn't a good crystal structure in humans. So that actually is probably better to use than the AlphaFold structure in those cases. So that's something we'd like to add in the future as well.
Any other question from anyone? And even after the webinar, if you think of any questions, there is a help desk for UniProt as well, I think. You can always send your question directly to UniProt team.
Yeah, and if it's related to ProtVar, it's better to send it to. We have our own help section. I mean, UniProt will probably forward it to us anyway. But if you specifically want help with ProtVar, then I mean, you can use the email I sent out already. But ProtVar does have one, so we can keep the questions together and we can answer them. And definitely, if you've got ideas, there's been some brilliant ideas today about things that you want to see in the future. So it doesn't have to be questions; it can be suggestions. Why don't you have this? This would be really cool. You should definitely do this thing in this visualization. Well, please send those as well, because having feedback from you guys is the way we can improve and help serve the community best.
Great. I think there's one suggestion already in. How well we can incorporate this mutation in the variants can be directly used to structure-based drug design.
Are you happy for me to take this one?
Yeah, sure.
Yeah, I mean, get the answer to that and you can make you $1 billion, right? And it's it's absolutely right. It's critically important to understand this. And I say the most direct way you could use what we already have is to look at, if from crystal structures, the contacts between small molecules and pockets, and then to overlay that with variants, either variants that have already been seen, and see whether they overlap. So I think this it's very interesting, but I think it goes beyond what we have in UniProt and ProtVar. I'd recommend you looking at Open Targets resource, because they have many more small molecules and drug informations linked with variants. So I would say looking at the Open Targets platform, we're more likely to be able to answer that question more closely.